the major objective is to process reads that come from a tissue or a collection of cells, so let's say cell culture or tissue, and then try to understand, typically at the difference between condition and control, what are some amplified pathways or gene sets. So that's the standard approach in RNA-seq, right? You kind of have to create a matrix of gene expression, levels, and then you can compare and contrast between different conditions. Now, in cell, what happens is that the resolution of your data becomes much, much more specific. And so I like this analogy that I heard, that instead of measuring temperature and averaging it across the whole hospital, right, where I can say I have dead people in the hospital, in the morgue, I have people with fever, very high fever, in maybe the infectious disease department, and I have average temperature maybe in other places as well. So if I measure the average temperature across all of these different conditions, I will get a normal temperature. But at single cell level, I am going to start to distinguish between the departments. And I can say, here's my acute patient department, here's the dead, and here are the normal people, right? So I can start to distinguish. And that is really one of the benefits of having such resolution. But how do we achieve that? How do we actually produce data that can help us see some of these differences? Well, in 2019, I believe this paper came out that kind of did a review of many different methods of single cell data preparation. And you will see here how first, these were very manual methods. So methods that people had to, take individual kind of several cells and try to separate them and so forth, and all the way to more and more automated methods. And so today, when we look at type of data that you are typically going to encounter, it comes from a specific machine that is designed to optimize, automate, and increase the number of cells and the accuracy of barcoding. But here you can see that, throughout from 2009, which the first paper that was published on single cell is in the middle of 2009, all the way to 2017, you can see an exponential growth of these single cell approaches. And so in 2017, actually, when this paper was published, they compiled all of these protocols that went from, very manual again to more and more automated. And you can appreciate now that whenever you take a data set that is single cell RNA-seq data, it actually could mean many different things. So to really appreciate the different approaches that have been more or less established over the past decade or so, maybe a couple of decades now, let's take a look at some of these basic ideas and technologies and try to understand how do we capture this precision and this dimension of single cell. So one of the first ideas is to differentiate between cells, right? If we are going to study something at the single cell level, we need to understand how do we differentiate between many thousands of cells that we potentially have to sequence. And to do that, one of the ideas is to give them a barcode. So if I could generate a barcode for a type of cells, right? I'm not generating a barcode for every individual cell. I'm just assigning them to maybe different tissues or different batches or something like that. So here I have barcodes. For example, I want to have a bunch of cells and I have a tumor and I have a microenvironment, right? And I want to be able to differentiate between them. I can give them a barcode, and those barcodes have to be encapsulated with the cell, right? And so if I kind of take a droplet, let's say an oil, droplet of oil, I can put a single cell into that droplet and a barcode bead. And that barcode bead looks like this. It actually is a bead that has these connected DNA strings attached to it. And once I prepare my data like this and I keep them all inside the same bead, now I can sequence individual cells. So this is the basic idea, right? But now let's take a look at three different methods I think I have here. So the first method is called Microwell. So the benefit of Microwell is that you actually end up with a plate, very similar to what you would do with microarrays, right? You have a plate, it has these small wells on the plate, and you're hoping to put in an individual cell within that microwell. Now, this is a process that could be optimized, and you can see in this case, this is that oil droplet, and here is that little cell, right? So that little cell right here, magnified over here, you can include additional phenotypic information. So essentially, in this case, you can make different color, fluorescence, and you can potentially identify an additional aspect just to give you a control, right? But in many cases, the limitation of this technology is that you only have a certain size, right? You can have limitless well plates, so you have a limited size. And so the number of cells is going to be quite small, right? So if we're talking about thousands, maybe hundreds of thousands of cells, you're going to be limited by the number of wells you have and the number of cells that you're going to successfully place inside each individual well. The second technique is called microfluidics RNA-seq. And so microfluidics really tries to separate everything, again, in a plate. So you have a plate, and that plate is where you put your cells inside this area right here. And then they start flowing over here. And as they flow through, you can see that you put in the cells, they start going in, and they get trapped. And so these traps right here is where you will end up having those individual cells. And those cells, again, are going to be, these are going to be cells that you would hope to then sequence at the end of the process, because then you can separate them. Okay, so this is that second option, right? When we have microfluidics. The final one, and probably one of the most successful ones, is the droplet single cell RNA-seq data preparation. And the idea here is that you have a continuous flow of drops. And here you can see that the cell and the hydrogen are incorporated into here. And as they flow, you can monitor them visually, and you can separate the ones that do have a cell in them or don't have a cell in them. So you have greater control over this. And you have here, you can see that it kind of encapsulates them into this oil bead, and then you can flow through and add the barcoding. So this is really a way to scale up and automate this whole process. And so the idea is that you encapsulate each individual cell inside a nanoliter droplet together with the bead. Then you can isolate the bead with the enzymes that are required to construct the library. And each bead will contain that unique barcode because this whole process is going to be automated, right? And at the end, you have this ability to separate your reads into individual cells, right? And this is the technology that is going to be used most often today when you encounter single cell data. For example, 10x Genomics is going to be utilizing something like this.